All right, today is our crash course review of biology. So recall the organization of the human body. We have the whole human body that is made up of the 11 body systems we discussed in our last lesson. And out of those 11 body systems, we're going to talk about many organs. However, those organs are made up of only 4 different types of body tissues, which will be the basis of our unit 2. And of those 4 tissues, our whole body is made up of trillions, about 30 trillion cells. So the 4 major body tissues are muscular, connective, nervous, and epithelial tissue. Next unit, we will go into deep detail about all 4 of these tissues. And then in the muscular system, we'll go into even more detail about the muscular tissue. And in the nervous system, we'll go over and go into more detail of nervous tissue. And in the integument, we'll go much more detail about the epithelial tissue. Muscular tissue is going to provide movement of the skeleton. It's going to provide movement of food through the digestive system and beating of the heart. So we'll learn that there are 3 types of muscular tissue. There's skeletal tissue, cardiac, and smooth. Connective tissue is the most abundant tissue in the human body, and it has a vast degree of functions. Connective tissue includes your bones, it includes your cartilage, and it even includes your blood. We'll talk about how blood is a liquid connective tissue, but it does meet all of the classifications of connective tissue. And if you think about the name as it implies, connective tissue connects things in the body like organs together. Nervous tissue is highly specialized, so it can actually conduct electrical impulses through that tissue. So this is going to include the brain, the spinal cord, and the nerves. While epithelial tissue is going to make up the linings of our body. So the skin, it forms a boundary between the inside of our body and the outside environment. The digestive tract is aligned with epithelial tissue. So food and substances don't actually get into the body until they pass through this layer of epithelial tissue. And it also includes our glands. So cells are made up of organelles. And one of the major organelles that we talk about in basic biology is that mighty mitochondria. This is that organelle that makes ATP energy through cellular respiration. Cellular respiration is a very common theme that we continually to refer back on, and the formula of cellular respiration. So we will be revisiting this mitochondria very often. There is also the endoplasmic reticulum. There are 2 types. There is the rough endoplasmic reticulum and a smooth endoplasmic reticulum. Those function for the modification and transport of proteins. We have the nuclear membrane that is going to have a barrier that keeps the DNA inside of the nucleus. And DNA has 2 forms. When DNA is in its loose form and not condensed into chromosomes, it is called chromatin. And then you have all of these other organelles like the lysosomes. We will talk a lot about those in the cardiovascular system during 202. And we talk about those leukocytes, how they destroy pathogens. A common structure we do talk about in all classes, though, is the cell membrane. And we want to recap and review the structure of that cell membrane. Then there is the Golgi apparatus, along with the nucleolus, where RNA is made, and those centrioles that have a big part in cellular division. You've got the ribosomes that are those little tiny red dots. Those are found throughout the cytoplasm and along the rough endoplasmic reticulum that makes it rough. The smooth endoplasmic reticulum has to do with the transport and modification of lipids. And then the cytoplasm is all that jelly-like substance that surrounds and supports those organelles. Here is just another view of a cell with all of those structures located, because the cell really is a busy place. The previous diagram shows a lot of space and a lot of that cytoplasm, but really the cell, it's pretty jam-packed with a lot of structures. So some structures that create kind of like a skeleton inside of the cell are these microtubules and the cytoskeleton. And then you'll see projections of the cell membrane that kind of stick out, and they make these little hair-like projections that are called cilia. And there is even smaller projections that can come off of the cell membrane, like the microvilli. And then some specialized cells, like sperm cells, will even have a tail-like structure called a flagella. So knowing the functions of the organelles is not required, but it is very, very helpful to know when clues are given within questions. So this is a great little slide just for you to recap what those organelles are and what the functions are. So when DNA is being used because the cell is doing its cellular functions, that DNA is in a loose, decondensed form called chromatin, and it kind of looks like a bowl of spaghetti noodles within that nucleus. Well, when the cell is getting ready to divide, it will condense its DNA into chromosomes. And chromosomes are really fun because you can actually count them. And human body cells have 46 chromosomes. So body cells mean everywhere in the body except for the gametes. So your hair cells, your eye cells, your skin cells, your stomach cells, your heart cells. They all will have 46 chromosomes that are made up of DNA. And those 46 chromosomes will be contained within the nucleus. So there's this extra room, safe room for protection. The gametes, which are the sperm and egg cells, those will have half of the genetic information. So they'll only have 23 chromosomes that are made up of DNA. And you'll talk more about those in 202. So as a cell grows, just like a child gets larger, so does the cell. It's going to start to get larger. However, it can only get so large before it's going to begin to divide. And the reason why that is because a cell cannot get too big. There's a reason why we're made out of 30 trillion cells instead of we're being just one large cell blob. As the cell gets larger, it becomes really inefficient at getting nutrients to the inner portion of the cell and getting waste removed from the inner portion of the cell and out. Because when nutrients are being moved in and out of the cell and waste, it relies on a process called diffusion. And diffusion is a passive process that takes time. So the larger a cell gets, it just gets too large for diffusion. So a cell will start to get bigger. And as the cell gets larger, it is going to continue to carry out its cell function. It is going to be in a phase called interphase. An interphase is the longest portion of the cell cycle. If you look at this little pie graph here, it's this whole orange phase on the outer part of the circle with the eye. And interphase can be broken down into 3 different parts. There is G1, S, and G2. So in G1, the cell is doing its cell thing. It is functioning how it should function. It is also getting larger in size and it's starting to replicate its organelles. Then it's going to go through a checkpoint and it will go into S phase. During S phase, this is where the DNA is synthesized or copied. So that way there are 2 copies of the DNA for 2 cells. Because that is ultimately the end goal, is to go from 1 cell to 2 cells. So then after that DNA is synthesized, the cell will go into G2 phase, where it will have a final checkpoint. If the DNA is mutated or there is something wrong with the cell is going to try to fix it. And if it can't, then the cell is going to be marked for destruction. And if it can, then the cell is going to fix it. As long as everything is good to go at the end of G2, the cell will enter into a phase called mitosis. And mitosis is going to divide the nucleus that now has 2 copies of its DNA. So the cell is going to divide on a regular cycle and it will prevent 2 genetically identical daughter cells, which is really important because we're trying to replace old or damaged cells. So if you have stomach cells that are damaged, you better be replacing those stomach cells with other stomach cells that can function as a stomach cell. So this is the process of mitosis. Mitosis is a process that occurs in the somatic or the body cells. And this is division of the nucleus. So you're going to have the DNA replicated during S phase of interphase. So here in this diagram, you have a cell with 2 chromosomes. Those chromosomes are replicated during interphase, right? But typically we see this in chromatin form. So it looks like loose spaghetti noodles. Then during mitosis, we are going to see those chromosomes condense. And now I have 2 copies. So that's why I'm going to see this X-shaped structure. It is the original chromosome and a replicated chromosome. So when the cell enters into mitosis, mitosis is going to be the division of those replicated chromosomes. And eventually you will have 2 copies of the parent cell. Each daughter cell has a copy of every single chromosome. So you can see here, these two daughter cells at the end have the same chromosomes as each other and as that original parent cell. So under a microscope, you can actually see these different phases of mitosis. And in order to see what's going on, you have to look at the nucleus and what's going on with the DNA. So if you look at this slide over here, it might just look like a big pink blob, but you can see this kind of boundary. That is the cell membrane. And then there's this little kind of darker purple structure. That is the nucleus, okay? So inside of the nucleus here, we're going to want to look at what the DNA is doing. And if you look at this DNA, I can't really see any type of X structure or rod-shaped structure. There's like just these like loose stringy things that kind of looks like a bunch of shoe strands or Christmas lights or spaghetti noodles, however you want to think about like a bunch of strings in there. So this is actually interphase. You can see that envelope of the nucleus, and you can see the chromatin. So during this portion of the cell cycle, the cell is performing its job. It's going through G1 where it's getting larger, it's synthesizing its DNA, and it is going through a checkpoint. Looking at this cell here, where the arrow is pointing, so here's the cell membrane, and there's this purple cluster, but the nuclear envelope has now disappeared, and we're starting to see these darker rod-shaped structures in the center of this cell. So this phase is prophase. The nuclear envelope is starting to disappear, and what we're seeing is chromosomes actually form. Once those chromosomes condense and form, the centrioles, though you can't really see them in the microscope, they are going to start to move to opposite ends of the pole of the cells. So you're going to have your centrioles here, and the other centriole is going to be here. And the chromosomes will begin to line up in the middle at the equatorial line or the equator. And so you start to see those X-shaped chromosomes line up right in the middle here. And you can see spindle fibers. Those are these rods that look like they're connecting to those X's. And they come from those centrioles. And as the chromosomes line up, those spindle fibers will connect to each one of those chromosomes. Once the spindle fibers have connected to those chromosomes, the centrioles will start to pull on the spindle fibers and draw them back into the centriole. Since they're connected to the chromosomes, those chromosomes are going to be pulled apart from one another. So you'll have one copy of the chromosome be pulled to one pole of the cell, and the other copy of the chromosome will be pulled to the other end of the cell. So we have these sister chromatids that make up the chromosome that are separating. So then, as the copies of the chromosomes get to opposite ends of the pole, now we're definitely seeing that there are two cells that are going to be formed. So now one copy of the DNA is on one side of the cell, the other copy is on the other side of the cell. So you're going to start to see a nuclear membrane that reforms around those sets of chromosomes. And you're going to start to see what's called the cleavage furrow. So that's this indentation. Because the next step is that the cell has to split the cytoplasm. So we'll go into what's called cytokinesis, where that cytoplasm splits and you have the complete formation of two cells. Meiosis is a process that produces sperm and egg cells, also called gametes. So this is a period of division in the gametes. And these cells start life just like other cells. They'll be in the cell cycle, where there'll be an interphase, and they will replicate each one of their chromosomes. However, something very special happens during prophase of meiosis 1. We have homologous chromosomes line up, and some of the information from mom's chromosome will switch places with the information from dad's chromosome. And what we end up with is that cell not just doing one round of prophase, metaphase, anaphase, and telophase, but it will do a second round without replicating its DNA. So after we do the second round of PMAT, prophase, metaphase, anaphase, and telophase, we end up with 4 cells that are genetically different or unique from one another and from the original parent cell. These chromosomes are made up of DNA, and DNA holds the code for how our cells behave and for our physical characteristics. DNA is said to be in the shape of a twisted ladder called a double helix. And the sides of that ladder are made up of sugars and phosphates. It's said to be a sugar-phosphate backbone, while the rungs or the steps are made up of nitrogen bases. And there are 4 nitrogen bases. There is adenine, thymine, cytosine, and guanine. And we have our base pairing rules, which state that adenine always pairs with thymine and cytosine always pairs with guanine. Adenine and thymine are connected by 2 hydrogen bonds, while cytosine and guanine are connected by 3 hydrogen bonds. This DNA is the code for all of the proteins that are required by your body. And there are 2 major processes within protein synthesis. There's transcription and there's translation. Transcription is where you take a molecule of DNA and you transcribe it or copy it into a molecule of mRNA. Think of like a scribe. They follow a doctor around and copy down everything the doctor says. So this is going to happen in the nucleus where the DNA is located. Translation is the second portion, and that's going to occur on a ribosome. That ribosome is either found on the rough endoplasmic reticulum or within the cytoplasm. And that process is going to take that molecule of mRNA and the ribosome will read the mRNA and use it as like a set of instructions on how to build a protein, and will call for specific amino acids so it can come in and start connecting them together to build that protein. Here is a great animation about how that process works from DNA to protein. Transcription occurs in the nucleus where DNA is located. And this is the process of converting the DNA code into an mRNA message. The DNA template's really easy to follow because we have those base pairing rules, right? A with T, C with G. However, with RNA, we have a substitute player. Instead of thymine, we're going to use a uracil instead. Translation is the second part. Once that mRNA molecule is made in the nucleus, that mRNA molecule, because of the uracil and being single-stranded, will be able to escape out of the nucleus and find its way to a ribosome. So once it heads to a ribosome, the ribosome will be able to read the mRNA sequence. And once the ribosome finds a start codon, it will begin to build a protein. And so the start codon on the mRNA message is an adenine, a uracil, and a guanine. And the ribosome will use transfer RNA molecule with an anticodon that has the appropriate amino acid on the other side. And it will bring in amino acid by amino acid, and the ribosome will connect those amino acids together. So these long chains of amino acids are called a polypeptide chain, which will eventually be folded into a protein. And we will pause here. I will pick up on movement of molecules and diffusion with the next video.